El programa de conservación de murciélagos les da un fraternal abrazo. Hoy iniciamos esta semana dedicada exclusivamente a charlas sobre los murciélagos. En la misma se irán tocando distintos aspectos de su biología, su ecología y su relación con la salud. Tenemos la dicha de iniciar con un referente en el estudio y conservación de los murciélagos. Es la curadora a cargo del Departamento de Mastozoología de la División de Zoología de Vertebrados del Museo Americano de Historia Natural y profesora de la Escuela de Graduados Richard Gere, del mismo museo. Sus investigaciones se centran en la morfología, sistemática y evolución de los murciélagos. Trabaja tanto con especies vivas como fósiles y está interesada en las relaciones filogenéticas, la biogeografía, la evolución de la diversidad ecológica y la estructura comunitaria de la fauna de murciélagos neotropicales. Morfóloga de formación, sus estudiantes y colaboradores la han llevado a muchas áreas nuevas de investigación en los últimos años, incluyendo la ecología de los murciélagos, las conexiones entre el genoma y el fenoma, los ectoparásitos y la microbioma que poseen los murciélagos, la dinámica de las enfermedades y la biología de la conservación. Es una de las principales organizadoras de la Unión Mundial de Redes de Diversidad de Murciélagos. También es presidenta del Grupo de Trabajo Mundial sobre Taxonomía de Murciélagos del Grupo de Especialistas de la IUCN y miembro de la Junta Directiva de Bat Conservation International. También forma parte del Comité Directivo de la Unión de Investigadores sobre Murciélagos del Sudeste Asiático, así como del Comité Directivo de Taxonomía y Colecciones del proyecto BAT1. Todo esto sin contar el gran número de estudiantes que ha asesorado y el gran número de publicaciones que tienen su apoyo. Sin más, les damos el inicio a la doctora Nancy Simmons, que nos estará compartiendo su presentación sobre los murciélagos, qué sabemos y qué no sabemos de ellos. Iniciamos. Nati.
Thank you. I will try to speak slowly tonight, but if I go too fast, hopefully someone will stop me. I'm looking forward to talking to you tonight about what we know and we don't know about bats. So some of this is very basic information, but some of it is new discoveries, new ideas, and where we're going as bat biologists in the modern age. I think everyone knows that bats are very diverse. They are found on every continent except Antarctica. And they range in size from very small, like this little bumblebee bat, to very large, like the big flying foxes. In terms of evolutionary history, bats appear in the fossil record about 52 to 55 million years ago. And some of the earliest fossils are the very best, with complete skeletons that are very clearly bats. So this is a bat that is 52.5 million years old. And you can see that it already has wings. And it is wings and flight that make bats really special. And it's from their wings that they derive their name, the order Chiroptera, which means hand wing in Latin. However, wings can be very different in different bat species, and the shape of the wings can tell us a lot about what bats can do. This bat at the top, Artibius, has what we call a low aspect ratio wing that is relatively short and broad. Comparatively, high aspect ratio wings are very long and narrow. And these differences in wing form have a large role in what bats can do in terms of maneuverability, where they can live, what they can eat, and how they fly. So bats with low aspect ratio wings tend to be slower flyers, but much more maneuverable. They can make sharp turns. Bats with very high aspect ratio wings like this are often very fast flyers, but they're not as good at making turns or maneuvering in tight spaces. So in bat faunas, we find that different species often live and fly and forage in different ways, depending on their morphology, their wing form, and also their diets. So in a single neotropical forest, we might find species, some of which live and fly near the ground all the time in dense vegetation, others that like to be higher up in the canopy, and still others that fly high over the forest. So a single piece of forest or grassland or savanna can support multiple different species of bats, each of them doing something slightly different. Bats, of course, are also known for their echolocation, making high frequency sounds that they use to detect obstacles, and in some cases to detect and track flying prey. The different sounds that bats make depend or are correlated with what they're doing. So a bat flying, searching, and trying to avoid obstacles may make one sort of call, and these are diagrams that have frequency on one axis, so how high pitched the sound is, and the length of the sound on the other axis. So this bat, while searching or just avoiding obstacles, makes one kind of call, but when it detects an insect, as it approaches and goes in to catch it, the call form changes. And this flexibility is important to bats being able to detect and track and catch prey on the wing. Interestingly, different species use sound and echolocation somewhat differently. Each of these abbreviations refers to a different species of bat, and these are their characteristic calls. So we can often identify different species based on the form of their echolocation calls when they are out searching for prey or approaching prey. So if we look at the big picture, echolocating bats that use sound this way are often called microbats or echolocating bats. There are 20 families, and about 85% of all bat species fall into this category. Another major lineage of bats are the Old World fruit bats. These are sometimes called megabats or flying foxes. And if you read in textbooks or general field guides, they may tell you that these bats do not echolocate, that they use vision instead. And in fact, they all do use vision. They have big eyes, which makes them look more like foxes or dogs. They all belong to one family, the Pteropodidae, and this is about 15% of all bat species. However, one of the things that we've learned in recent decades is that some pteropodids, the flying foxes, actually do echolocate. Recently, a paper was published, actually several papers, showing that some pteropodids are actually quite sophisticated at echolocation. We have known since the 1950s that some pteropodids, the members of the genus Rosetus particularly, click their tongues and make clicking sounds when they're in dark spaces, like in the backs of caves. But Yossi Yovel and his colleagues showed that these bats actually receive very detailed information from these tongue clicks. We used to think that these clicks were very primitive, that might tell a bat when it was about to hit a wall, but not much else. But it turns out that they emit the clicks from different sides of their mouth. So the right side of the mouth, then the left, the right, then the left, the right, then the left. And by alternating the click emission pattern, they can get, by listening with the ears on each side of the head for the difference in when the sound comes back, they actually have echolocation, which is almost as good as all other bats in terms of helping them detect things and navigate. So it is not so primitive after all. These bats do echolocate. They've also found another form of echolocation in the fruit bats, the old-world fruit bats, and that is clapping their wings together. So the tips of the wings, they hit together like clapping. These bats use the returning echoes from those wing claps to get information that helps them to navigate. So it turns out that the old idea that some bats echolocated and the pteropodids just didn't do it is wrong. Well, what about diet? And what about using echolocation to catch prey? Well, at least 80% of bats use echolocation to help catch arthropod prey, like this moth being chased by this bat. It turns out that this is one of the things that bats do that make them most valuable to humans. Now, bats are valuable in so many ways to ecosystems, but when one is trying to communicate to the public, to a farmer, to somebody you meet on the street, the value of bats, it is interesting to know that we think that $53 billion in non-toxic pest control per year is given to humans by bats. So this is an important figure. These are important informations, particularly in this time that bats are being vilified. And this figure comes from just a few months ago. $53 billion in pest control of value to agriculture. But of course, bats eat many, many things. And when I first started studying bats, I thought that all they ate were small insects like moths. And I was amazed to discover that they actually take all sorts of other prey, very large katydids, grasshoppers, and even centipedes and other large arthropods. I do not have a picture of a bat eating a large spider, but they do that too. Still other bats are true carnivores in that they eat other vertebrates, frogs, mice, birds, and even other bats. And some, of course, also are fishing bats. So these bats are found in the Dominican Republic. They eat small fish when they can get them. Then there are the vampire bats. So many people, the public often thinks that vampire bats are everywhere and that they're very large, but this is the foot of a chicken. So this tells you the body size of a vampire bat. There are actually 3 different species of vampire bats that have evolved. They all live in the neotropics, although not out in most of the Caribbean. So they're found in Central America and South America. They're all small, and they feed on the blood of birds and mammals. Still other bats are frugivores. They eat fruit. Sometimes they eat relatively small fruit for their body size that they can carry and eat whole. Sometimes they eat large fruits where they eat the pulp and drop the seeds on the ground. But in all cases, they're important for seed dispersal and forest regeneration. Still other bats feed from flowers. They may eat the flower parts, but often they simply eat the nectar and the pollen from flowers. Here is a classic neotropical nectar feeding bat visiting a banana plant. And they're very important in pollinating bananas and many other plants. These bats often have long snouts, long noses, and they have very long tongues that can extend down inside flowers. So that's what's happening here. The bat has stuck its face into the flower and its tongue is reaching down into the flower for the nectar. But when they approach flowers, of course, they get the pollen all over themselves. And so they can be very important for pollination of many plants, including plants of agricultural importance. All of these plants, for example, these fruits, which are raised and sold by people. Durian in particular is very important in Asia. Bananas, of course, around the world. Many fruits here are pollinated by bats. Now, all of these different adaptations to different diets are reflected in the morphology or the anatomy of the bats, particularly in their skull forms. So these bats represent the diversity of diets in bats. This is a carnivore. This is Vampirum spectrum that eats mice and birds. This is a big fruit-eating bat, Artibius. And this is a nectar bat with the long rostrum for reaching into flowers. Other bats down here are small insectivores and so on. You can often tell the potential diet of a bat by looking at its skull and teeth. However, in recent years, we've discovered that these easy divisions into feeding guilds, saying, for instance, that this bat is a nectar-eating bat, or another bat is a carnivore or an insectivore. It turns out that the bats often have much more varied diets than we thought from watching them a few times or looking at their skulls. For example, this bat, Glossophagus soracina, was always categorized as a nectar bat, but it turns out when people started looking at the detailed diets from studying the DNA in their fecal samples, that they eat all sorts of insects as well. So this bat sneaks an insect a snack, as this title of this paper shows. This is just one example. There are many examples where we are finding that bats do not know what guild we put them in. In fact, they eat many more things than we thought. The same holds true for the carnivorous bats. We used to call these bats carnivores. Many people knew that they also ate insects. But we assumed that the small insect bats like this one, this is a Micronycterus, known for eating insects that it gleans from surfaces like this. We always thought this is too small, this bat must only eat insects. But doing long-term detailed studies of diets, Charlene Santana and her colleagues found that in fact, these little bats are carnivores when they can be. They're very small, so they're not big enough to take many sizes of prey, but when they can, they eat small insects. And they actually filmed them catching eating lizards. They actually filmed them catching and eating small lizards. So this bat is not an insectivore strictly. It eats what it can catch, which is usually insects, but is sometimes vertebrates too. We are also learning a lot about the different places that bats roost and how important this can be to their ecology and their ability to live in different places. Everyone knows that bats live in caves. I think practically every child, if you ask, where would you look for bats, would say a cave. But bats live many other places as well. Tree hollows are common roosts for bats. And actually in this tree in Belize, I and my colleagues took over 100 vampire bats out of this single tree in one night. So the tree is hollow up inside and hundreds of bats can live in a big hollow tree like this. Bats also live inside fallen trees. So when a big rainforest tree comes down and rots out in the middle, bats may live inside those hollows. They may also live in burrows in the ground. And this is an armadillo burrow that bats previously, we didn't know where they lived. It turns out this is the kind of place they go. They may also live under banks and along streams. And some of these we've discovered actually like man-made roosts, like inside culverts, like these man-made tunnels. So under a road, you may have a metal tube that looks much like the burrow of an armadillo or another large animal, and bats may roost there. This is a Maya ruin, a Maya temple that looters dug a hole inside. It's like a cave. Bats like to live there too. Some bats, however, live in the open. These are proboscis bats, which are common in many neotropical areas, and they roost in this characteristic pattern on trees over and branches overhanging water. And they're quite well camouflaged. You can see they blend in nicely with the bark. Other bats live in unmodified foliage like this under palm fronds. Here's a white Diclodurus, a ghost bat that's roosting up here. Other bats may roost under almost any palm frond, but some actually make tents. So this is a leaf tent made by a small fruit-eating bat where they chew around the edges of part of the palm frond to make it sag down. And then they can live underneath that saggy part and be protected from rain and predators and things like that. Other bats live in dry foliage. Sometimes they like thatched roofs of people's houses. They also live under bark. Finally, this is one of my favorites. In places that have termites that make nests up in trees, some species of bats, particularly bats of the genus Tonacea, chew holes in the bottom of termite nests and live up inside those holes. The termites wall off the hole where the bat has chewed and make a nice, safe cavity for the bats to live. More interesting in some ways from a biological and conservation perspective is recent discoveries we've made that, or observations rather, that bats can be some bats are very particular about what kind of roost they use and will only live in one place, where other species may be more flexible. So at the field site I work at in Belize, we have bats that the same species may occur inside a tree hole, inside holes in the ground, and inside man-made caves. And what's important here is that species that are more flexible may be more likely to survive and thrive in forest fragments, where many kinds of roosts may be absent or less frequent. And we found in this fragment of forest in Belize that there were certain species that tended to still be present in this fragment as opposed to a larger piece of forest. And those were the roost generalists, species that used more than one kind of roost. So looking at roost ecology can sometimes give you important information that can help understand why some bats may survive in fragmented habitats and other bats may not. So how many bat species make up a local community? Well, the answer to this depends on where you are on the planet. In temperate areas, like in North America, bat faunas consist of maybe 20 species in one area, but somewhere between 0 and 20, for instance, in most of the United States. But as you approach the tropics, species diversity increases enormously. And this is true on each of the continents. Africa, the numbers here are quite wrong because there hasn't been that much work done in Africa at the time that this map was drawn in 1993. We did not know as much. But if we look here in South America, over 120 species may live in the same area. And then that number goes down as you go towards the poles in each direction. Apparently, largely an effect of latitude due to different food resources and roosting resources. Well, what about the Caribbean? Current estimates are that there are 79 different bat species that live in the Caribbean. And if we look more closely at the island of Hispaniola, there are 18 bat species found in Hispaniola. Now, this may not sound like a large number, but it is actually quite a diverse fauna. And it's not just important because of the diversity of the animals, but also that there are several species found only on Hispaniola and nowhere else in the world. One of these is Molossus virilii. This used to be thought to be part of a widespread species, Molossus molossus, but was recently shown to be a separate species. And I was unable to find any photographs of Virili. This is actually a picture of Molossus molossus. They are similar, but it would be great to get a good picture of Molossus virili. Pteronotus pusillus is another species found only in Hispaniola. It's endemic to the island. It used to be thought to be part of the widespread species Pteronotus parnellii. Now we know it to be a different species, found only on your island. Nautilus major, a third one, used to be thought to be part of Nautilus terminius. Now we know that it is a distinct species. So how many bat species are there in the world? Well, this is a moving target, but we can go back and look. In 1982, Carl Koopman, who was my predecessor at the American Museum, published the bat chapter in Mammal Species of the World, the first edition. And he recognized 918 different bat species. 10 years later, a few more bat species. Koopman, 1993, recognized 925. Well, unfortunately, Carl died in 1998, and I inherited the job of keeping up the list. And I thought it would be a very easy job. It turned out to be much more challenging than I thought. And in 2005, in the last edition of Mammal Species of the World, I recognized over 1,000 bat species. So this seems like a big increase. In that edition, for every bat species, we included the name of the species and the authorship for that name, the common name of the bat, type locality, distribution, conservation status by the IUCN Bat Specialist Group, synonyms, so those are names of both subspecies, the ones here in bold are subspecies currently recognized, and then comments of where to look in the literature. Well, how many bat species are there compared to other mammal species? Well, through these three editions, 82, 93, 2005, the numbers kept going up. But interestingly, the percentage of mammal species that are bats stayed more or less the same. And that's because in other mammal species, we were also identifying new species through each of these iterations. So what comes next? Well, this book was published in 2005, and that is 15 years ago now, which is a long time. So where are we now? Well, we're now in the modern age of the internet, and rather than having the bat list stay out of date, I began working over 10 years ago with Andrea Cirinello, one of my colleagues, to put the bats of the world online in a database where you could obtain all the same information that was in the original books, updated. And so we've been working on this now, and it is available at a website called batnames.org. And so this is freely available. Any of you can go there anytime. By using the explore and query functions, you can look up any taxon you want or any geographic area you want and find information on the bat species. It's been a lot of work, and we do not have complete accounts for everything published online yet. So for some species, you will only find the name and authorship and not all of the information on synonyms. But we have that, and we're working on getting it all up for people to use. To make all of this more of a community effort and less of something that I do by myself with Andrea, in our kitchens, we founded the Global Bat Taxonomy Working Group in 2017 to bring together a group of taxonomists to help make decisions about what taxa to recognize. So the goal was a community-based approach to updating and stabilizing bat taxonomy. For easy taxonomic new descriptions of species, we can just add those to the database, but sometimes there are complex problems that need multiple taxonomists to look at them and make a decision. And that's what this group is for. The Global Bat Taxonomy Working Group now has, I think, 18 members, different countries around the world. We are a little North American and European heavy because there's a lot of taxonomists there, but we have also been adding taxonomists from Asia and Africa and so on. And ultimately, we hope as new young taxonomists get interested in these kinds of questions of diversity, we hope to add more people to this working group. The group is an official group of the IUCN. We are part of the IUCN Species Survival Commission Bat Specialist Group. So this is the umbrella under which we work. So again, a reminder, any of you are encouraged to go use the bat names database anytime you have questions about bat taxonomy and distribution. So how many bat species are there in the world? Well, as of March of this year, our latest update, 1,421. This is more than 300 species additionally added to the list since the 2005 volume was published. So that's a 27% increase in the number of recognized bat species in the last 15 years. Let's look at a map and think about where these new taxa are from. These are the additions to the list since 2005. 17 new forms recognized in the Caribbean, 26 in Central America, 80 in South America, and so on. As you can see, in every part of the world, we're recognizing new species. And these are the total numbers. So the largest number of species in any geographic area is Southeast Asia, with nearly 400 different species. But there are many islands here, and so there are many islands endemics. Just as there are many island forms in the Caribbean as well. Fewer in North America, 48 species total, over 300 in South America. Worldwide, 22 additional bat species have been added every year over the last decade. Actually, I think this number is a little higher now. It keeps going up. So this is per year we see this amount of change. Of these, roughly 60% are new species. That means species that need a new scientific name, that there's not already a name for that animal. 40% are taxa raised from synonymy. So these are most often groups that were thought to be a subspecies of something that are now considered to be distinct species. So these are the cases of the endemic species from Hispaniola, where all had scientific names before, but they were thought to be just subspecies. Now they're recognized as distinct species in their own right. If we look at the pace of discovery, and I think this is an interesting thing to think about, we look at the number of valid species described per decade over time, and a few things stick out. First of all, in the early part of the 1900s, a whole lot of new species were described. And this is really the age of colonialism, the age of discovery, when big expeditions from Europe were going out and cataloging new organisms from different parts of the world. Then after that age, things sort of settled down. And so we had, through much of the 1900s, sort of an even keel pace of new species being described per year. But then it started to go up in the 1990s and 2000s, going up really fast all of a sudden. So what caused this? Well, I think it's a combination of things. First of all, exploration of new places. Finding new species often comes with going to places where nobody has ever gone to study bats before. but also new technologies, particularly the technologies relating to bat detectors and using acoustic monitoring to look for bats. And remember, I said that bat echolocation calls were often distinct by species. Well, sometimes people going into a place will find calls that they don't recognize, which will be a clue that maybe there's a species there that no one's ever studied before, noticed was there, and maybe a new species that hasn't ever been seen by scientists before. However, the biggest revolution has really been in the use of molecular techniques, DNA analyses, looking at genes of bats to decide whether two animals are the same species or different species. And so much of the revolution, and particularly the recognition of species, pulling them out of synonymy, saying this subspecies is really a distinct species, come from detailed studies of molecular work. And I think you're going to hear more later this week on these topics. But it's also study effort. How many people are out there studying bats? This is another graph I put together a long time ago because I was just curious. How many papers on bats appear per year in the literature over different decades? And I just used Google Scholar to do this. In the 1920s to 1950s, roughly 20 to 25 papers on bats were published each year. That means one person could sit down and easily read them in a couple of weeks. And then that number started to go up in the 60s, in the 70s, and 80s. So I started working on bats in 1989. And during the early years of my career, it seemed that there were lots of bat papers coming out. That was about 550 per year during the 1990s. But since then, it's just gotten crazy. Since 2010, over 2,000 papers every year published on bats, and the numbers are just going up. Let's look at the most recent year that we have complete data from Google Scholar. About 3,500 papers on bats were published last year. If you search on Chiroptera plus ecology, you find that about 2,400 papers were published just last year alone on bat ecology. 2,000 on bats and conservation. Bats and disease. I want to spend a little bit of time now. This is what I'm sort of getting near the end of my talk, talking about, this is on everybody's mind now. What about bats and disease? Well, over 1,000 papers were published on bats and disease last year. But it turns out that most of them were about white nose syndrome, a disease that bats get, a fatal disease that is an emerging infectious disease in North America. These are bats, hibernating bats, that are suffering from white nose. You can see the noses look oddly white. That's where this disease gets its name. It's actually a fungus, a cold-loving fungus that grows on bats while they're hibernating. And you can see here the effects. It covers all parts of the skin of these bats, and it causes lesions. So this is a skin infection that's caused by a fungus called Pseudogymnoascus destructans, or P. destructans as it's called. And you can see all of these wounds or lesions in the wing of this bat. What this fungus does is it disturbs the bats while they're hibernating and continually wakes them up. And they burn up the fat reserves that they have to stay alive through the winter. So when they run out of fat reserves, there's no food around because it's winter, there's no insects, and it causes mass mortality. This is a heartbreaking picture from a cave in the northeastern United States. Hundreds, thousands of dead bats at the end of the winter that basically died from white nose syndrome. This is a disease which was unknown before the winter of 2007. And it was first identified in a single set of caves in New York State, 3 caves that first winter. And then it spread. So The blue was the next year, and then the green, and so on. And so now in the most recent winter that we have data for in this map, we have white nose syndrome, all of it spreading in the Western United States as well. Every year it moves, it covers more range. So there are now 13 bat species that are affected with white nose syndrome, which is the syndrome that kills them, where the fungus grows actively in their tissues, and 8 additional species that have been found with the fungus on them, although not actually suffering the disease to the extent that it kills them. These 3 species, which are all small insectivores, have experienced over 90% mortality, which means that their populations are just decimated to below 10% of what they were before white nose came. So one of the things that's most shocking about white nose syndrome is where we think it came from, which is humans brought white nose syndrome to North America, probably on contaminated boots or caving gear. The fungus is very closely related, if not identical, to a fungus that naturally occurs in Europe. So European bats are not affected by it the same way because they evolved with the fungus, but it is something that was introduced in North America to a naive set of populations of bats, and it has been devastating. So that now brings us to bats and COVID-19, which has been on everyone's minds. So this is a different example. Obviously, white nose syndrome is a disease that humans brought to bats, while COVID-19 is a disease that many people think may have originated in bats and is causing a lot of concern in the conservation community because of misconceptions that people have about this link. So the disease, the human disease COVID-19, is caused by a coronavirus called SARS-CoV-2. So SARS-CoV-2 is the virus, COVID-19 is the disease in humans. The virus has been found to be 96% similar to a coronavirus that was found in a horseshoe bat in China in 2013. So horseshoe bats are the genus Rhinolophus, which is widespread in the old world in Africa and Southeast Asia and Asia. So let's look at that 96% similar figure. Well, if we look at whole genomes, the human genome and the chimpanzee genome are 95% similar. So, and here's the reference for that figure of 95% from 2002. So 2 organisms that are 95% similar are very different biologically, as different as humans and chimpanzees are. So the fact that the bat virus and the human virus are about this different means they're actually quite different viruses, even though they are related. And so this is a phylogeny showing SARS and SARS-CoV-2. Here is the human SARS virus, the original SARS virus, and all the gray boxes are bat viruses. So it's clear that the human SARS virus originated in a clade of viruses found in bats. Down here, this is SARS-CoV-2, which causes COVID-19, and the most closely related virus is also a bat virus. So we have, this is the bat virus, which is found in Rhinolophus, and here's the human virus, SARS-CoV-2. These 2 viruses shared a common ancestor, which, based on what we know about the rate of change in viruses over time, suggests that the common ancestor of these 2 viruses existed somewhere between 1950 and 1980. So that's somewhere between 40 and 70 years ago. So the bat virus and the human virus are not the same. They've in fact been separated from 40 to 70 years. Where has the virus been during this time? Well, it could well have been in some intermediate host, like the pangolins, which have somewhat similar viruses, maybe a carnivore, or maybe in another bat. We just don't know yet. And so saying or implying to people that they might get COVID-19 from a bat is simply wrong. We do not know where the virus which causes the human disease has been for the last 40 to 70 years. And this is one of the things that researchers are studying actively at the moment. However, this mere mention of bats and COVID, the fact that the virus probably originated sometime in the past in bats, has caused a great deal of backlash against bats. In some parts of the world, people are actively killing bats because they think that they're going to get COVID-19 from the bats, which is just not true. If you're going to get COVID-19, you're going to get it from other people, not from bats. Yet this backlash exists and is very dangerous for bats. There's also concern that bats may be able to get COVID-19 from people. That is, that humans, bat researchers, or people exterminating bats may come in contact with bats, and if the person is sick, they might transfer it to the bat. And this is based on some cases where humans have clearly given COVID to other animals. For instance, at the Bronx Zoo in New York, the tigers got COVID-19 from their human handlers. There's another case of fur farms of mink getting COVID-19 from the people working at the fur farm. So there is concern that it might be possible to give COVID-19 to bats. And this actually led to the IUCN Bat Specialist Group and other groups around the world basically arguing that it's recommended to suspend field activities that involve handling bats until we learn whether it is actually possible to give the disease, transfer SARS-CoV-2 to bats. And this is being studied in a number of different laboratories now. Just a few days ago, so I guess this was last week, the US Geological Survey published a large document assessing the risks posed by SARS-CoV-2 to North American bats. Part of the concern here is that white nose syndrome has already decimated bat populations in North America. What would happen if we gave these highly threatened bats yet another disease? This is a bad thing, obviously. So basically, being very careful is being recommended for all bat researchers around the world. Finally, one of the last things that's come out of this whole thing with COVID is mobilization of a new network of networks of bat researchers and conservationists called GBATnet, the Global Union of Bat Diversity Networks. And I'm one of the organizers of this group. We have long planned to bring together all of the bat research and conservation networks around the world into some sort of more unified whole where we could learn from each other. This has been discussed at several international bat meetings by the leaders of many global networks. But in April of this year, we decided that because of the bad press that bats were getting, that we needed to do this sooner rather than later. So GBATnet has a big social media presence now. On Facebook, we have our own Facebook page. We also have our own Twitter account. And the Twitter account here identifies the reason for having GBATnet, which is to bring together, actually this number is wrong, it's now 14 existing bat networks for exploring complementary aspects of bats to facilitate and enhance research addressing bat biology and conservation, particularly sustainability, protecting bats for all time. Now, as I said, this is a network of networks, and the local network for those of you in the Dominican Republic and Latin America is RELCOM. So this is a very active regional network which covers Latin America and the Caribbean. And so RELCOM is one of the participating members of GBATnet. What GBATnet does so far is to try to disseminate information coming from our networks to get them to other groups around the world. For example, here's a post of a recent publication on guidelines for communicating about bats. So if you are asked by people in the street, your friends, your family, or journalists in your town about bats, should we be afraid of bats? Why are bats important? What do you know about bats? This publication contains useful information. So we advertise this to various members of our social media to provide help. We also put, anytime there are good media summaries of information useful to for bat education and communication, we post these in our social media. I'm biasing myself here because of my age. I am much more comfortable with Facebook than Twitter, but we have all the same things and even more on Twitter. We also have a series of expert blogs. We have a blog spot where we post articles that are written to contribute to the community. Here's one on human impacts and the spillover of zoonotic diseases that we posted back in April. Here's another one. Killing bats won't help prevent disease, but here's what will. So we get experts who are able to read the literature and find all the references to write summaries that anybody can read. And in this case, all of what's in yellow here gives you links to the original scientific papers if you want to go back and find more information so you can teach your class about it, or just because you don't quite understand it yourself. I use these summaries all the time before I have to have interviews with journalists to make sure that I have my facts straight. So this can be very useful. If you want to participate in GBATnet, the best way to do this is to participate in your local network, RELCOM, and help RELCOM and volunteer your time and your efforts there, because RELCOM is one of the contributing networks. You can also, as an individual, join the Facebook and Twitter. You can follow us on these social media, and then as an individual, you'll get all of these posts and you can see all of this information yourself. And with that, I would like to wind it up and thank the organizers for inviting me to give this talk. I'm not sure how we're going to handle questions, but I'll leave that up to you. So Natalia, if there are questions coming in Spanish, I don't know if you can speak so that I can hear or somebody else can connect us.
Sí, ¿cómo te ayudo? Wow.
Okay, nice. Hold a second. We go to another person to do the question for you.
Okay.
Okay, Amel.
Miguel, apague lo del intérprete para que podamos hablar sin eso.
Natalia, ya. Tú puedes hacerle las preguntas. Está en tu WhatsApp. Tú lo apagaste el intérprete, Joel.
Ah, pero no, tan solo fue para mí, aparentemente.
Ya la quité. Listo. Listo, quité el intérprete. Háblale, Natalia. Intérprete.
No.
Listo.
Okay. Can you hear me now, Nancy?
Yes, I can hear you now.
Okay, fine. Okay, so yes, I have a couple of questions. Do you know anything about the existence of the fungus in the Caribbean, of the white nose, the cause of the white nose syndrome?
I do not believe that anyone has found the fungus that causes white nose syndrome in the Caribbean. That would be a big concern, but the fungus affects, it's a cold loving fungus. So it affects bats when they're hibernating at lower temperatures. So I suspect it would not be fatal for bats in warmer climates anyway. So it's a concern for its fatal effects only in northern climates where the temperatures are lower that encourage the fungus to grow.
Okay, I'm gonna jump in with a question of my own on the topic. I work with plants, so I know very little about bats, but I can assume that bats in the Caribbean don't hibernate, so it wouldn't be a concern. Okay.
Right. Yeah. So there are some bats that they go into torpor. So it's a much shorter period of time. So some tropical bats will let their body temperature go down when they're not active, but it doesn't go down enough or for long enough for the bats to grow fungus, even if they have it.
Okay. So there's another question. So based on your knowledge, because the geography. Oh, I'm sorry. Let me translate to everyone else. Si bien ella dice que como el virus afecta a los murciélagos que están hibernando, y de hecho en el Caribe las especies de murciélagos no hibernan, no se ven afectadas. Entonces ella dice que me explicaba que en el Caribe y que en el trópico hay algunas especies que entran como en un periodo de hibernación corto, no sé si se llama tupor, si es el término correcto en español, pero que incluso a estos no les afecta, no hay evidencia de que les afecte el virus. Okay, so another question is, based on your knowledge, because the geography of Hispaniola is so varied from elevations that go up to 3,100 meters, and it only has 20 species. Do you believe that this number is an underestimate of bat diversity on the island?
I don't know the answer to that. It depends. It's always possible to find something new in particularly places that no one has looked before, where the habitats are different. So maybe very high elevation sites, there might be something new. But I suspect that all of the species that are known from the island, I guess I would be surprised if there were new ones, but you never know. And also hurricanes can displace species from other islands. And so it's always possible that new species will arrive at any time. So you never know.
Ella dice que no tiene la respuesta exacta a esa pregunta. En resumen, ella piensa que no deben haber especies sin reconocer de la española, que siempre es posible, que es posible que en las elevaciones, en las altas elevaciones, es posible que hayan algunas especies a identificar, pero que le parece poco probable. También dice que los huracanes pueden traer especies, o sea, individuos que estaban en una isla pueden ser traídos por los vientos hacia otra isla, entonces siempre es posible que aparezcan nuevos registros en las islas, que se muevan de esa forma. Okay, there's another question. After every night of work in the field, we watch with chlorine the bags where the bats are kept. Now we see that we also have to take care of the clothes. Any other recommendation for biosafety regarding bats? This is from Luis Avila in Mexico.
I think that going forward, most bat researchers, maybe all bat researchers, will start wearing masks when handling bats and stop breathing and blowing on bats. Wearing gloves, and possibly eye protection, wearing something to cover your eyes. But if we are concerned about infecting the bats, the most important thing is covering the nose and mouth so that we're not breathing on the bats.
Ella entiende que en el futuro los biólogos trabajarán con mascarillas y guantes.
Dile la pregunta también, por favor.
Oh, I'm sorry. Perdón, no estaba diciendo la pregunta. Preguntaban que luego de cada noche de trabajo lavamos con cloro los sacos donde guardamos los murciélagos capturados. Ahora vemos que el cuidado de la ropa debe ser considerado. ¿Alguna otra recomendación para la bioseguridad de los murciélagos? Ella dice que en el futuro ella entiende que los biólogos tendrán que ir con mascarillas y con guantes a trabajar en el campo, en particular si están preocupados por la seguridad de los murciélagos y de no infectarlos, que lo más importante va a ser cubrirse la boca, no respirarles encima a los murciélagos mientras de trabajo. So another question. Thank you very much for the talk. Wait 1 second. So do you know what were the changes, the morphological changes in the carpal region of the bats in relation to other mammals?
Wow. So the carpal region is the wrist, and the wrist bones have been rearranged in bats somewhat. The same bones are there, but they're arranged in different ways. One bone, the pisiform bone that is in our wrists, you can see it on the outside, is moved to the ventral surface in bats. And the action of the wing is in sort of a different, it's restricted in some ways where we can move our hands in more directions than a bat can move its wing. So they are constrained to make flight work, but there's a whole series of changes in different bones in that area.
Bien, la pregunta es, ¿se conocen cuáles fueron los cambios morfológicos en la región carpal de los murciélagos en relación con otros mamíferos? Ella dice que, bueno, que la región carpal es el área de la muñeca y que tienen los mismos huesos que el resto de los mamíferos, pero que la posición ha cambiado. Entonces ella mencionó uno en particular, pisiforme, no sé si no es así que se dice en español, pero que nosotros lo tenemos, se ve de este lado y los murciélagos como cambió hacia el otro lado y eso hace que el movimiento se restrinja de una forma particular y así puedan volar. Y que así otros huesos también han cambiado en su forma para permitir el vuelo. So another question is, what is your opinion on the importance that genetic has right now over morphology in the process of discovering or naming new species? To what point is genetics able to answer questions about new species?
Genetics or genetic tools are amazingly important for recognizing new species because they give us information we could not get any other way. However, it's important to have more than one line of evidence when deciding whether two populations are one species or the same species. So, for instance, using just mitochondrial genes, I don't think is enough because mitochondrial genes are inherited through the maternal line, and sometimes it's the males that are doing all the dispersing. So I like to see data, mitochondrial genes and nuclear genes and morphology and echolocation calls. So the most secure assessments of new species are based on multiple lines of evidence. But one of the most important, or two of the most important, or three if you include sex-linked genes too, are the genes, because they get us information that we cannot get other ways. And they have been enormously, and will continue to be, one of the main tools that we use. That said, sometimes you catch a bat and you look at it and you go, this is a new species. If you're you know what the diversity of species look like. Sometimes new species are very morphologically distinct and recognizable immediately. So sometimes all you need is morphology. But then when you do the genetics to back it up, you find that they're also very different genetically. So the best thing is to have multiple lines of independent evidence before making up your mind about whether something is a new species or not.
Bien, la pregunta es, ¿cuál es su opinión sobre la primacía que se le ha dado a la genética por encima de la morfología en el proceso de nuevas especies? ¿Hasta qué punto la genética logra responder las preguntas sobre nuevas especies? Entonces, ella dice que la importancia de los análisis genéticos es importantísimo porque da mucha información que no se puede obtener de otras formas. Pero a ella le parece que el mejor enfoque es tener varias líneas de evidencia. Entonces, tener datos genéticos de varias formas, por ejemplo, datos mitocondriales, datos nucleares, y eso acompañado con información morfológica. Por ejemplo, ella decía que no tener solamente información del ADN mitocondrial le parece muy insuficiente para determinar si dos cosas son diferentes o lo mismo. Porque solamente se hereda de la parte de la vía materna y muchas veces son los machos los que se están dispersando. Ahora bien, a veces una persona que conozca bien la diversidad de murciélagos puede ver un murciélago en su mano, tenerlo ahí y darse cuenta de que es una especie nueva. Ahora, entonces se le hace la genética, entonces también se observa que son diferentes genéticamente y eso hace una descripción o una identificación mucho más robusta. Entonces ella recomienda que haya diferentes líneas de evidencia genética, de mitocondrias, del núcleo, datos morfológicos, etcétera, para tener una descripción. Okay, so
that was a long answer.
That's okay. Let's see. Okay, I read that one. How many fossil species of bats are reported?
So fossils can range from beautifully preserved to little fragments, a few teeth. Altogether, the last time I counted, there were somewhere between 200 and 300 different species that were known from fossils. Some of those seem very similar to living forms. So, you know, probably somewhere between 1 and 200 actually named species of fossils exist, of bats in the world.
Bien, la pregunta es, ¿cuántas especies de fósiles de murciélagos hay reportadas? Entonces ya hace la salvedad de que los fósiles pueden variar significativamente, ser por ejemplo una especie con un individuo completo preservado o algunos fragmentos de dientes. Entonces ella dice que la última vez que revisó habían como 230, 230 you said?
Somewhere over 200, yeah. But some of those are assessed to living species, yeah.
Bien, como por encima de 200 especies registradas de fósiles, pero que ella dice que algunas de esas son muy similares a especies que están vivas ahora mismo. Entonces ella cree que entre 100 o 200 especies fósiles se conocen. So bats are very important in the Caribbean for seed dispersal. Which methods do you use for studying these?
For studying seed dispersal, if you can catch the bats, you can collect fecal samples. I have a graduate student who's been studying bat diets by DNA barcoding their feces. and finding all sorts of surprising things, that fruit-eating bats eating a few insects, insect-eating bats eating all sorts of things. For the plants, you can sometimes get whole seeds frequently get whole seeds out of fecal pellets. The problem is that some fruits are so big that the bats don't ingest the seeds, that they just chew the meat off the fruit and then spit the seeds out. So, so if you can catch the bats and you can get the poop that way, that's a good way. Another method is to put out tarps under known roosts of bats, and then you don't actually have to catch the bats. You can just collect the poop and study that. So that's a non-invasive way of studying what the bats are eating and how they're dispersing the fruit.
Bien, la pregunta era sobre los métodos para estudiar la dispersión de semillas de los murciélagos. Entonces ella explica que en el mejor de los casos se puede capturar el murciélago y analizar las heces fecales. Entonces a las heces se le pueden hacer estudios genéticos para determinar cuáles especies están presentes, cuáles restos genéticos de las especies están presentes en las heces. Que en el caso de los murciélagos que consumen frutas puede ser un poquito diferente. Las frutas con semillas pequeñas, los murciélagos se la comen y eliminan las semillas en las heces. Pero hay veces que cuando la semilla es muy grande, esto no es posible. Pero que también otra forma de hacerlo es poner como unas carbs, no sé el nombre como en español, pero como algo abajo de donde los murciélagos perchan para que para que las heces caigan ahí y no tener que colectar el animal. Okay, so another question. Do you think U.S. protocol for WNS survey, I don't know, does that mean something?
White nose syndrome survey.
Okay. Survey would work for bat survey in COVID-19 times, because that protocol involves the use of mask and alcohol disinfection.
I think that is the way things will go, is that we will use much more PPE, personal protective equipment, to protect people, but mostly to protect the bats. The problem is there's a shortage of all of this kind of gear right now because medical workers need it. And so there's also an ethical consideration. Should we be using masks and respirators and, nitrile gloves and Tyvek suits and stuff that medical people need to handle bats? I don't know, but I think recommendations will be coming in, upcoming months. But the short answer is we're going to have to be much more careful than we used to be, and probably very rigorous protocols to be extra careful.
Bien, la pregunta es si ella considera que el protocolo que se usa ahora mismo para trabajar con murciélagos, que se utiliza para el síndrome de nariz blanca, que sí sería útil en los tiempos ahora de COVID-19. Porque esos protocolos requieren uso de máscara y alcohol y eso. Entonces ella dice que le parece que esa es la dirección en la que se va, que la gente tendrá que usar mucho más guantes y mascarillas y cosas de eso, pero que ahora mismo en medio de la crisis hay también una cuestión de ética, que es que esos materiales están escasos y el personal médico lo necesita para trabajar. Entonces, que no se debe utilizar ahora en esos momentos, pero que ella cree que esa es la dirección en la que va. Que sabe que vendrán recomendaciones próximamente, pero que le parece que esa es la línea. Do you know bibliography on the protocols that will allow us to take DNA from collected individuals and deposit it in museums?
So yes, there are sets of recommendations of best practices for collecting samples, both non-lethal samples, like where you take a punch of wing tissue and then you let the bat go to collect genetic samples that way. Also to collect whole animals, to collect organs for real genomic approaches. And the person to contact for that information is Liliana Davalos, because she's helped develop a lot of that information for the BAT1K project. And I think she's going to be speaking later in the week. But it's not hard to collect tissue samples, but it's also important to put voucher specimens if you do sacrifice an animal, if you euthanize an animal and kill it to collect tissues. It's very important to make sure that all of that gets into a museum somewhere so that it can be used by future researchers. So I published a paper back in, oh gosh, I don't know, 15 years ago on how to prepare specimens and take very simple tissue samples, but there's been many advances for DNA collection since that time. So I can provide a reference on how to prepare a specimen of a whole bat for a museum. But for the tissue samples, you need more recent, I think, references for that.
Natalia, perdón, creo que la pregunta estaba enfocada a los especímenes que ya están colectados.
Oh, the question was focused to specimens that are already collected and deposited in museums.
Oh, for already collected?
Yes.
So people frequently are getting good DNA out of a variety of parts of museum specimens. So sometimes even from ones that are preserved in alcohol. But for a dried skin, you can get tissue from the lips or from the incision where they opened the animal up before stuffing it with cotton, or from clips of the skin. From fluid preserved specimens, sometimes tissue samples from deep inside give good DNA from muscle tissue and things like that. So it depends on what the goals are of the study. It's easier to get samples that produce good mitochondrial gene results than it is for nuclear genes, because there's more copies of mitochondria in each cell. So it depends on what you're trying to do, whether you can use a museum specimen easily or whether it's going to be much harder or impossible.
Bien, la pregunta es si conoce bibliografía de protocolos que nos permitan tomar muestras de ADN de individuos colectados y depositados en los museos. Entonces, ella dice que actualmente cuando se trabaja con especímenes de museos se toma ADN de varios tipos de muestras que puedan estar preservadas en alcohol, que puedan estar secos, etc. Y que depende el objetivo, o sea, el tipo de ADN que uno quiera extraer, si funciona bien los especímenes de museo o no. Ella dice que es mucho más fácil si obtener muestras de ADN mitocondrial porque es mucho más abundante en las células que de los genes nucleares. Entonces eso va a ser un factor importante, pero que sí que se puede sacar, que por ejemplo se saca mucho de los labios y algunas zonas donde se cose cuando se va a preservar la piel del animal. Do you know anatomical descriptions of the, wait. Do you know anatomical descriptions of the carpus region of the fossil species? Like, for example, of, and I'm not going to say this well in English, Onychonicteris. I don't know. Okay.
Yeah.
I don't know if I translated the question correctly. Did you get something?
Well, unfortunately, we do not have a lot of information about that region in a lot of these fossils, so the descriptions are partial. Whoever it is, tell them to write to me and I will see what I can do if I can point them. There are some good descriptions of some fossils that region, but I don't know about that one. I don't recall it being well preserved.
Okay, la pregunta era si se conocen descripciones anatómicas de la región carpas de las especies fósiles, como por ejemplo Deoniconicteris. Entonces, ella dice que de esa región se tienen algunos, pero pocos registros, que ella me parece que dijo que de ese género como que no se tiene mucha información, pero que a la persona que hizo la pregunta, que le escriba en privado o le mande un email y ella puede darle más información detallada. Another question. Taking into account your experience, what do you think is the most remarkable variable that could make a difference to roost selection by bats on temperate and neotropical areas?
I'm not sure I understand the question. I think different bat lineages have evolved to take advantage of different kinds of spaces as roosts. I don't think there is a single variable that is important across all the different types of bats and the different places that they live. We know that temperature and humidity and light levels are very important. Other than that, we don't really understand why certain species roost in certain places and others don't. That's an unanswered question, I would say. And it's probably very complicated, as many things are like that.
Bien, la pregunta es que si ella cree que hay una variable más importante que determine la diferencia entre la selección de perchas de murciélagos en zonas templadas y en zonas neotropicales. Ella dice que no cree que haya una variable en sí más importante, que ella cree que la temperatura, la disponibilidad de luz y la humedad son factores determinantes en la selección del área de percha, pero que esta realmente es una pregunta que todavía no tiene respuesta, que no se sabe bien por qué una especie elige un lugar para perchar sobre otra. Another question. Biosafety should be both ways. Is it possible for bats to transmit viruses to scientists handling them?
Yes, rabies is one example. We always have to be aware that if you mishandle wildlife of any kind, it's possible that you could be exposed to something. In any place where rabies is possibly present, bat researchers should not handle bats unless they have had rabies prophylaxis shots. It's just basic safety. So one has to be aware that this is a possibility. However, any of us who have been in the field for many years have handled hundreds, thousands of bats with no gloves. We've been bitten by hundreds of bats. And if it was easy for us to get viruses from bats, we would probably all be dead. So spillover from wildlife to humans is not necessarily easy. And I don't feel that I have put myself at risk by handling bats over the years, but I will be more careful going forward because safety goes both ways. And as we learn more, we realize that we have to be more careful than we were in the past. I don't feel that bats are dangerous to me. I intend to continue handling bats once we learn more about the whole COVID problem, but I think I will be more careful in the future, and I think I will be wearing gloves in the future and things that I didn't necessarily do in the past. So it's a matter of risk assessment and making good decisions. So, yeah.
Bien, la pregunta es, la bioseguridad va en ambas direcciones. ¿Es posible que los murciélagos nos transmitan virus a los científicos que los manejamos? Entonces ella dice que sí, que en particular, por ejemplo, el caso de la rabia, que cualquier biólogo que vaya a manejar murciélagos en un lugar donde la rabia sea prevalente, no debe manejarlos a menos que no tenga los las inyecciones profilácticas contra la rabia. Entonces, ella comenta que en su experiencia de campo, ella ha manipulado miles y miles de murciélagos, la han mordido cientos de murciélagos, y si ella piensa que si fuese tan fácil que los murciélagos le transmitieran enfermedades a los humanos, entonces todos estaríamos muertos. Más sin embargo, ella dice que ella tendrá en el futuro más cuidado de no manipular los murciélagos sin guantes y sin mascarillas para disminuir la probabilidad, pero ella no le tiene miedo a los murciélagos, ella va a seguir trabajando con murciélagos y no cree que el trabajo directo con estos animales presente un problema significativo. Did the common ancestor of bats have the ability to fly?
I would say what we know today is that, yes, the common ancestor of all living bats was definitely a flying mammal, as were some of the close relatives of that clade. So we have ancient fossil bats, which are not inside the smallest group that contains all the living bats. So we have successive sister taxa, which were clearly flying animals. The most basal member of the bat lineage that we know of is the fossil Onychonicterus, and it seems to have been able to fly but not echolocate. So we think that flight evolved before echolocation in bats.
Bien, la pregunta es, ¿el ancestro común de los murciélagos tenía la capacidad de volar? Entonces, ella dice que sí, que definitivamente se cree que el ancestro común de todos los murciélagos sí podía volar, y que además lo que sí creen que la ecolocación echolocation evolved after flight.
Yes.
Es cierto que la ecolocación evolucionó luego de desarrollar la capacidad de vuelo y que incluso muchos grupos cercanos a los murciélagos también tenían capacidad de volar. So what is the most recommended method to euthanize bats in case it's necessary?
Well, what I prefer to use is as an anesthetic that veterinarians use for, dogs and cats and things. It's called isoflurane, and more or less it's something that bats can inhale and then they fall asleep and then they die when you overdose them on it. So it takes only a minute or two and it's painless and it's very quick. So if one can get access to a veterinary anesthetic like that, that is by far the best way. However, other traditional ways of euthanizing bats include one thing is called cervical dislocation, where you basically break the neck of the animal very quickly and they die. It's not much fun. Another way is basically thoracic compression, where you compress the chest so they can't breathe and then they suffocate very quickly. Yeah, I know, it's awful. I've done that many times. I don't like it. And so I'm very happy using isoflurane now.
I'm so happy I work with plants.
Not everybody has access to it. So these other methods are very quick because bats have a very high metabolic rate. And so they have very rapid respiration and very, very rapid heartbeat. So I believe, they basically black out and die very quickly. But still, I prefer to use isoflurane or another veterinary anesthetic if I can get it.
Yeah, so happy I work with plants.
Yeah. But bats are so wonderful though.
La pregunta era que cuál es el método más recomendado para eutanasiar a los murciélagos en caso de que sea necesario. Entonces ella dice que el mejor método es un anestésico que utilizan los veterinarios para gatos y perros, y isoflurane.
Yeah.
Isoflorano.
Isoflorano, gracias Miguel. Que eso los murciélagos lo pueden inhalar, se duermen y mueren rápidamente luego de que se les da una sobredosis, y es rápido y no sienten dolor. Que si no hay el anestésico se pueden hacer dos cosas: o romperle el cuello o presionarle el pecho, hacer unas compresiones para que dejen de respirar. No le gustan esos dos métodos, pero que son muy rápidos. en caso de que no se tenga el anestésico, que definitivamente ya prefiere trabajar con el anestésico, pero que ha utilizado los otros métodos cuando no lo tiene disponible. So the last question is, what is your personal motivation to study bats?
I started studying bats just because I thought they were really cool. I just thought they were really interesting, amazing animals, and I was stunned that there were so many different kinds and they do so many different things. And so I started working on bats because I was interested in all those ways, and I never expected to keep working on them, but almost any interesting biological question about animals you can address using bats as a study organism because they're so diverse and they do so many amazing things. So I'm just, every year I'm even more interested in bats. There's just so much to do with them and so many unanswered questions, and they're just really cool. They're just wonderful animals.
Bien, la pregunta era que ¿cuál es su motivo personal para estudiar los murciélagos? Ella dice que comenzó a trabajar con ellos porque le parecían muy chulos y que nunca se imaginó que iba a pasarse el resto de su vida trabajando con ellos, pero que cada año que pasa le gusta más, que le encanta que todavía hay tantas preguntas sin responder, que le parecen organismos muy interesantes y que sigue muy apasionada por los murciélagos. So I've been asked to say thank you so much for everything. It was a pleasure to hear you speak, and that the guys here hope that they will get to meet you in person one day.
I hope so also. That would be great. I'm very excited that there are so many people interested in studying bats in Dominican Republic and elsewhere in Latin America. They're, the more the better. So I'm very happy to help out.
Bien, ella dice que le encanta saber que hay tantas personas interesadas en los murciélagos en el Caribe, en República Dominicana y en Latinoamérica en general, y que también espera poder seguir colaborando y conocer a todo el mundo en persona en algún momento.
Amelia.
Estoy aquí. Bueno, queremos darle las gracias a todas las personas que han participado en Zoom, en YouTube. Queremos darle las gracias por todas las preguntas que han enviado y por participar durante todo este tiempo con nosotros. Nos disculpamos por cualquier tecnicismo con la tecnología que haya ocurrido. Solamente recuerden que mañana pueden utilizar el mismo link que fue enviado de Zoom, el cual se encuentra también en Facebook, pueden acceder a él. Recuerden que tienen que tener ya una cuenta en Zoom para poder acceder, y luego que se envíe ese correo es que a usted le va a llegar el link para que entren directamente y participen en Zoom. Sin embargo, vamos a estar en vivo en YouTube, así que no se preocupen. Recuerden que las preguntas de YouTube también fueron respondidas por nuestra panelista y conferencista el día de hoy. Así que nos vemos mañana. Recuerden que tenemos la presentación de Ángel Soto Centeno el día de mañana. Así que nos vemos mañana a las 7 de la noche.